Okay, welcome to Level 1 Science Online with Henny van Vieren. Hi, you 11s. Welcome back to another online lesson. Hey, back to reality. Anyways, we need to continue learning about genetic variation. There's quite a bit to get through, and we only have about, I want to say 3 weeks, maybe a little bit more to get through all of this. So, we are going to have to pick up the pace. I'm really sorry that I couldn't be there last week. I had pneumonia, and consequently, I'm hoping that you all tried your best to get onto Education Perfect. We have to get ourselves ready for the practice exams, and this is going to be in your life probably the most important practice exam that you ever do, because if we go into lockdown during the actual NCEA exams, your practice exam grade will count. So we really need to make sure that we can do this. Okay, so jumping right in, just a reminder, what we did work on in the past was all about genetic variation, and we talked about chromosomes and alleles and DNA and how it all fits together. So in 2013, question 2a was use the diagram below to help explain the relationship between chromosomes, genes, alleles, phenotype, genotype, and the molecule of DNA. Now, can you see this is an excellent question, guys? So you need to basically learn how to tell the story that a chromosome, you start with DNA. DNA is a long molecule with a sugar phosphate backbone. It is a double helix and it is made up of nucleotides. The sequence of the nucleotides, or the order of the nucleotides, contain all the genetic material of the body. The way that it is stored is by basically being twisted and twisted and compacted, and the compacted form of DNA we call chromosomes. Now, on a piece of DNA that codes for a specific trait or gene, well, for a specific trait or protein is called a gene, and consequently, so it's a section of DNA that codes for something specific, but we get two chromosomes from sexual reproduction. So one chromosome was from our mom and one chromosome was from our dad. And both of these contain the gene for eye color, which is that specific section of the DNA that codes for eye color. But the differences between them, we call them alleles, because one was maternal and one was paternal, unless they are obviously the same. But if they have difference, it's the same gene, but the same gene for eye color in this case, but the alleles are the two different variants or the two different forms of that same. Your phenotype is your physical feature, and your genotype is the combination of your alleles, whether one is dominant or recessive, and that actually, your genotype determines your phenotype on how the offspring will look. So all of that I just said, you need to be able to explain exactly the story. Okay, so I'm not going to go through all of this. I actually expect you to go through all of it. Make sure that you can do this. Okay. Please remember that if you are talking about alleles, then you have to refer back to the example. So if they are talking about eye color, refer back to eye color, please. Okay. Now this was in 2014, so you're supposed to know the structure, and if you look at it, AGCT in the diagram, now AGTC at the Gold Coast. Alright? So we know that A goes with T, and we know that G goes with C, and we know that T goes with A, and C goes with G. Okay. Sorry about that. And then you're supposed to know that this is a phosphate, and that is a ribose sugar. And this is what we call a nucleotide. And these things that keep them together, your two phosphate sugar backbones, which is part of your helix, they are called hydrogen bonds. Okay, so there's all our answers. Now again, question B over there was the relationship between DNA, a gene, and an allele. You see it's the same as the previous one, just a little bit simpler. Now, you are not expected to write down thymine, cytosine, adenine, and guanine. You can only remember that it's T, C, A, and G. Okay. So, in 2015, a snail known as Sepia nemuralis can have either a plain shell or a banded shell. Okay, so this is obviously turbo, it's the one that goes faster, and there's the plain shell. The diagram below shows the homologous chromosomes that contain the gene for the shell pattern for each of the snails in the photograph above. So, assume the allele for the plain shell is dominant over the allele for the banded shell. Okay, so normal is dominant on striped. So when you look at a homologous pair of chromosomes, is one from the mom and one from the dad, but they contain exactly the same proteins. Okay, so you need to know what a homologous pair is. So the homologous pair in this case, one was from the mom, one was from the dad, and as you can see there, there is the gene for shell, okay? And then the allele for the plain shell is dominant, and you can see that this snail has got both the dominant and the recessive alleles. So when we know that we have a dominant, it means our phenotype will be the dominant phenotype. And the recessive can only show recessive if both are recessive. So once you see somebody with a snail with a shell with a pattern on it, or the stripe on it, then you know that it has to be little n, little n. Okay, so that's the introduction to the question. Then, in the diagram on the previous page, which snail is heterozygous for shell pattern? Hetero, remember, hetero means more than one, homo means same, same. Okay, explain why you chose this snail. So, this one is heterozygous because it has two different alleles for the same gene. Then, that was for achieved. Now, referring to the examples shown previously for the shell pattern, explain the difference between allele and a gene. So again, same thing. What is a gene? What is an allele? What is a chromosome? What is DNA? All of that. Okay, so these are the rock pocket mouse. Rock pocket mouse can have dark fur or light fur as shown below. Using the example of the rock pocket mouse fur color, explain how information carried on the DNA controls the appearance. In your answer, you should refer to DNA base sequence, genes, and alleles. And then color is going to be your phenotype, and your genotype needs to be in there. Okay, but they want you to specifically say two different alleles have different base sequences. And if they have different base sequences, that makes them code for different things. Okay, so specifically the base sequence. And when you talk about base sequence, you always need to talk about the complementary base pairing rule. Okay, please go and read the model answer. So here's the thing you need to memorize, basically, this slide. Okay, so chromosomes made up of DNA. DNA is a large molecule that is coiled into a double helix twisted ladder. It is responsible for determining the phenotype or the physical feature of an organism. Along this molecule, the DNA, are bases and nucleotides. These bases pair up A to T and G to C. Okay, that is complementary. A sequence of bases which codes for a particular trait, something like eye color, is called a gene, and genetic information is determined by the order of those bases. So if the bases have different orders, you will have a different trait. Different versions of each gene are called alleles. These show the different variations. Because chromosomes come in pairs for each trait, there will be two possible alleles. Remember, two chromosomes means two different versions of the same gene. Now, these different versions of genes occur as the DNA base sequence is different. The combination of alleles for each trait is called the genotype. This can be any combination of two available alleles. Genotype is the combination of the alleles. This determines your phenotype. Okay, whichever allele is present may be expressed. So dominant, when it's there, it will be expressed over recessive, and recessive can only be seen when there's only recessive. Okay, so that was, here we go, so genotype and phenotype. The allele for brown eyes is dominant over the allele for blue eyes. Discuss how it is possible for a child to have blue eyes even though both parents have brown eyes. In your answer, you should use labeled Punnett squares. Link the genotype and phenotype of a child and the grandparents. So, first thing we need to say is somebody with brown, because brown is dominant, brown can be either big B, which is homozygous, or it can be heterozygous, which is big B, little b. Now, in this case, the child has got blue eyes, and we know that blue is recessive, so it has to be little b, because if there's a big B, it will be brown. So the child has to be little b. And for the child to actually get a little b from the mom and a little b from the dad, it means that they have to be heterozygous, which means both parents have big B, little b. They cannot have big B, because the moment one can only give a big B, then it's a problem. So let's see how they answered it. Yeah, so the grandparents could have genotype. Oh, use label pun and link the genotypes and phenotypes of the child, parents, and grandparents. So both parents have brown eyes, so therefore they both must have a dominant allele. And because each parent passes on a recessive allele to the child, they have to be big B, little b. Now the same with the grandparents. To have a child that is big B, little b, the grandparents could have a genotype of little b, that could be grandma, and grandfather could be big B, or big B, big B. Okay, so, but to create that, you need a combination of these. It's not possible to say for sure, but at least one of the grandparents on each side must pass on the recessive allele. In order for the two parents to have a recessive allele, Punnett squares may be used to show and is suggested. Okay, here we have another one. Two brothers who have the same parents and they are not identical twins will have different genotypes and phenotypes. Define the term genotype, define the term phenotype. Explain how the two brothers with the same parents can have different genotypes. In your answer, you should explain the importance of meiosis and the role of fertilization. Okay, we'll get to that part later. But genotype, phenotype, you can see there's another important definition. Okay. We're back to the snails. So the one with a stripe and the other one not. So these two snails were produced by sexual reproduction from male and female. Discuss how they have inherited different alleles for shell pattern. In your answer, you should explain homologous chromosomes, where they come from, and give possible genotypes for both parents and explain how you determine these possible genotypes. Okay, exactly the same question as the eye color one. Okay, so make sure you read these yellow blocks. They are really important. Okay, then back to the rock pocket. Dominant is dark fur is dominant, and light fur is recessive. So each mouse has two alleles for fur color. Explain how they inherit these alleles. Explain how two alleles interact to produce the phenotype. In your answer, you should define phenotype and genotype. It's exactly the same, guys. Fur color, blue eyes, striped shell. It's exactly the same. Explain how these alleles are inherited from the parents. And state the three possible fur color genotypes for rock pocket mouse, which is homozygous recessive, little b, heterozygous, which is big B or big D, little d, and homozygous dominant, which is big D, big D. Okay, so Punnett squares. We use Punnett squares to keep track of alleles when calculating the genotype of any offspring. So if you look at this, B now is the dominant allele for the agouti color. I imagine that's the agouti color. So the moment it has a big B, it's going to have that color. The little b is a recessive allele for black color. So the moment we see a black rabbit like this, it is going to be little b, little b. It has to be. Because if it has a big B, it's going to show the agouti color. So when this rabbit makes sperm or egg, it's going to split those into two, and one will go, basically it can give a little b or a little b, or it can give a little b and a little b. Because remember, it's got two chromosomes, and they are homologous pairs. So when he was made, he got one from his mom and one from his dad. Same as this one. Got one chromosome from his mom. On that chromosome is a gene, and that gene is the color. Got this chromosome from the other one, the mom or the dad, and the allele to the fur color. So in this case, we use the Punnett square, and we can have big B down there, and little b across. So in this case, this is how we determine the genotype, and 50% of the offspring will be agouti colored. Okay? And that is for every fertilization. It doesn't mean if they have 4 kids, and 2 will be one color and 2 the other. No, it is for every fertilization. So every time this guy makes sperm or an egg, and every time this guy makes a sperm or an egg, and they come together. Okay, so Punnett squares, that's how we predict the probability of having different phenotypes. Here we have lop-eared. So if you look at, it seems like if we use the example of straight and lop-eared rabbits again when they breed, and all the offspring will have a genotype RR and the phenotype of straight ears. If we cross 2 big R little r's, or heterozygotes, the genotype and phenotype ratios of their offspring second generation can be set out as below. So if you have somebody that's heterozygous, they currently have straight ears. It means that the straight ear is dominant and the lop ear is recessive. Then this guy gives, in production of his sperm or egg, he gives a big R, that's the one chromosome, gives that same one for the second option, and in this case gives the small r and the small r. This guy, same thing. So you will have basically a homozygous dominant, that's over here, and a heterozygote, there's 2 of them, and then a homozygous recessive. Now that's our genotypic ratio, but our phenotypic ratio says if it's got one dominant, it's going to be straight ears. So can you see there's 2 straight ears there and 1 straight ear here. So they're going to ask you, write down the genotype ratio or the phenotype ratio. And the way that we write them down is by using a colon. So 1 to 2 to 1. Can you see it's 1 to 2 to 1. Okay, so, and they are always written in that order. 1 to 2 to 1. Okay? So that is 1 dominant homozygous, 2 heterozygous, and 1 homozygous recessive. You need to say what they are. So 1 to 2 to 1. 1 dominant homozygous, 2 heterozygous, and 1 homozygous recessive. If they ask you the phenotype ratio, in this case it will be 3 purples to 1 white. 3 purples to 1 white. Okay. Okay, I think that's where we need to stop for today. I will post the pages that you need to do for me in the site pad. Okay, thanks guys, thanks for your time, and thanks for your hard work. Good luck out there. Remember, you're not alone. If you need to talk to somebody or need some help, please reach out to me. Okay? Look after yourselves, please. Thank you. Bye.